Hello, welcome back to my biology class. In the previous class, we were discussing on the historical developments in the field of microbiology. So in that, I think we have discovered how microscopes were developed and what are the organisms were observed and who is the father of microbiology, why we call him as a father of microbiology. And also we discussed on spontaneous generation theory conflict, who proposed the spontaneous generation theory, and who are all the scientists who have tried to disprove the spontaneous generation theory or theory of abiogenesis. And we have also studied how Louis Pasteur, by a simple and unique experiment, disproved the spontaneous generation theory. So in this class, we will continue to study some other important findings or developments in the history of microbiology. So you know now who is Louis Pasteur. So Louis Pasteur is a chemist. Originally, he is not a microbiologist. He is a chemistry professor in France. So on the request of his friend, so he has studied on, okay, so on actually spoilage of wine. Okay. So Louis Pasteur is a great scientist in the field of microbiology. He has contributed immensely for the development of microbiology in the mid part of 19th century, mid to later part of 19th century. So we will let us know, so what are all the developments or findings that were made by Louis Pasteur. So as I was telling in the beginning, Louis Pasteur is actually a chemist. He is not a microbiologist. And in Europe, especially in France, so there were many wineries which are present, so which were producing wines for more than 1,000 years. But in 19th century, so these wineries means the factories which produce wine from grapes. So the wineries were facing some problem in wine production. The problem was the wine used to get sour. So ಹುಳಿ ಆಗ್ತಿತ್ತು. So when wine used to become sour, they used to throw it off. So like this, many wineries were suffering huge losses because they had to throw large batches of wines. So because it has spoiled. So one of the Louis Pasteur friend, he also had a winery. So during that time, so only newly the presence of microorganisms were discovered, and but the wine was prepared or preparing in wineries for more than 1,000 years, and they were thinking that the production of wine is a chemical process. It is not a microbiological process. Therefore, so Louis Pasteur friend requested a chemist who is a Louis Pasteur, who is a chemist, to see what is the problem, why the wine spoilage is happening, and he has also requested to give the solution to overcome the wine spoilage. So what Louis Pasteur is done is, he has simply taken two samples of wine. One is a good wine, another one is a spoiled wine. One is a good wine, and the second one is a spoiled wine. So during that time, microscopes were there, the presence of microorganisms was identified. So Louis Pasteur had an idea of that. Louis Pasteur, what first he has done is, he has taken the two samples, one spoiled and one good wine. He has taken the samples of that on the slide and observed under the microscope. So when Louis Pasteur observed this under microscope, so he found one type of microorganism present in good wine and two types of microorganism present in the spoiled one. So out of the two types of microorganisms that were present in the spoiled one microorganism was similar to that of the microorganism present in the good wine. So next step, what he has done, he has isolated two types of microorganisms present in the spoiled wine and separated and pure culture them. And he has also isolated the one type of microorganism that was seen in the good wine. So now he has got two types of microbial culture from spoiled wine and one microbial culture from the good wine. So next what he has done is in the next step, he has taken the fruit juice, he has boiled that to kill all the present microorganisms. Then he cultured these three microorganisms which he has pure cultured and inoculated to the three flasks containing sterilized fruit juices, grape juices. So after fermentation, he kept them for fermentation. After fermentation, so when he tasted the wine, so one wine was sour or spoiled, and other two wines were good. So other two microorganisms formed a good wine. So with that, he identified that the one microorganism present in the spoiled wine was similar to the one which was present in the good wine. So with this, so he identified that the other microorganism which is causing the sourness is, he identified that as a lactic acid bacteria, and he said that the contamination of fruit juice with the lactic acid bacteria is causing the sourness or wine spoilage. Okay, and he has also said that the other organism, he named it as yeast, and he said that the formation of wine is a microbiological process and it is mediated by microorganism called yeast. That is, he said that yeast are responsible for the production of alcohol and wine. Apart from that, he has also given one solution. He has also given one solution to overcome the wine spoilage by lactic acid bacteria. What he has suggested is, he has suggested pasteurization. So initially, pasteurization was given or started with to avoid the wine spoilage. So what he suggested is, he suggested to heat the grape juices and then inoculate with the yeast. So when you heat the fruit juices, it is going to kill all the contaminating microorganism. And when you inoculate with the culture of your interest or a good yeast, so you will not get the contamination in wine or sourness. So like this. So he has done many discoveries in this simple experiment. First thing, he identified that lactic acid bacteria causes wine spoilage. He identified that wine production is a microbiological process. He identified that yeast are responsible for production of wine or alcohol. And he has also identified or given a technique of pasteurization to kill this wine spoilage microorganism. So initially pasteurization was developed to kill the contaminating microorganism in fruit juices or in grape juice before using it for wine. So here he suggested to heat the fruit juices up to 60 to 62 degrees centigrade for 1 to 3 minutes. So he has not suggested to heat more because heating the liquid containing sugars more, so it will spoil the characters or the quality of the fruit juice. So this is one of the contribution of Louis Pasteur. So and he has developed pasteurization. Now the pasteurization is also used to kill the contaminating microorganisms or pathogenic microorganisms in milk. So it is in all dairy industries. So pasteurization is done for milk. So pasteurization is done at two temperature. So one at 63 degrees centigrade, another at 71 degrees centigrade. That we will study in industrial microbiology. So he proposed pasteurization, that is heating of the liquid, not up to boiling, below the boiling point to kill the spoilage and pathogenic microorganisms in 1864. So he also demonstrated that many microorganisms are the causal agents of many diseases. Or he said that bacteria can make the human being sick, or bacteria can cause the disease in human being. And in 1860, here it is given 1857, but it is around 1860, he proposed germ theory of diseases. What germ theory? Along with the Robert Koch. So what germ theory of diseases says is, some diseases are caused by small organism. Some diseases are caused by small organism which are too small to be seen with a naked eye. So the growth and multiplication of these organisms inside the human being, plant or animal causes the diseases or makes the human being sick. So that was proposed by Louis Pasteur in 1860. That is called germ theory of diseases. So small organisms which are too small to be seen with the naked eye will enter into the human body. The growth and multiplication of these organisms inside the human body will make the human being sick or diseased. That is called the germ theory. Proposed, that is the germ theory of diseases, says. So, but yes, during that time, he has not given the proof that which organism will cause which diseases. Though the proof for germ theory of diseases was provided by Robert Koch. We'll study that in the future slides. Yes, you may be knowing that he has also developed vaccination for rabies virus. And he has also identified lactic acid bacteria from curd. And he said that the storing of milk or formation of curd from milk is also done by microorganism. So, and the organism involved is lactic acid bacteria. And apart from that, Louis Pasteur also identified the causal agent of silkworm febrile disease. Pebrin, febrile disease in silkworm, identified that this is caused by Nosema bombycis, a protozoa. Nosema bombycis. Okay, these are some of the contributions of Louis Pasteur. Now, we'll study about another scientist called John Tyndall. So, John Tyndall, you know that in bacteria, so there are several types of resting stages or different groups of microorganisms form different types of resistance structures or resting stages under unfavorable conditions. For example, if the temperatures are very high, if the temperature very cool or cold, or if there is no presence of any moisture, no presence of any nutrition, at that time the microorganism may die of extreme environmental conditions or by hunger. So to avoid this, the microorganism have developed a mechanism. What they will do is they will convert it to another structure, a resistant structure in which it can able to survive for very long time. So in this way, the fungi produces fungal spores. Bacteria produces different types of structures. In that, endospores is one type of resistant structure. Cyst are one type of structure produced by bacteria and also protozoa. So among all these structures, the endospores produced by some group of bacteria called Bacillus clostridium, they are highly resistant to adverse environmental conditions. So these endospores can survive for years together. Even if you keep that endospores in a boiling water for 24 hours or 1 month, it will not affect anyway. It will still survive. So to destroy, so if that survives, what will happen is that will keep contaminating in microbial culture, wherever you want to culture. So if you are not killing that one, that will grow it, if there are any presence of moisture and nutrient. So to avoid that one, one scientist called John Tyndall, he has given one heating method to kill the resistant spores of or endospores of bacteria. So what he has proposed is, so if there is any nutrient media which you have prepared or any liquid or any solid material, so what you have to do, what he has proposed is heat that to a 100 degree or heat that to a maximum temperature for some 1 hour, then allow it to cool. Allow it to cool for 1 day. For example, it is a broth, culture broth. So heat that to 100 degrees centigrade for 30 minutes, allow it to cool down and leave it, cover it with a cotton plug, then leave it for 1 day. So what will happen here? So when you heat it, all the vegetative form means those forms which are in viable or which are multiplying, which are having an active growth cycle. So which are viable cells. So they will get killed if you eat above 60 degree or 70 degree centigrade. So only remainings will be cysts and spores. So when there is a nutrient media, so nutrient rich media is there. So when there is no competition because you have killed all the growing cells, so then these spores or cysts, what they will sense is, so there is a favorable environment, there is a presence of much nutrients, so no competition. So they will come out of spore form to vegetative forms. So what Tyndall said is, after eating for sometimes boiling, so you heat it again after leaving a gap of 24 hours or 1 day. Again heat it for some time. Boil it for another half an hour. So when you boil it next time, what will happen? All the endospores might have converted into viable vegetative cells and they also get killed. And he has suggested that leave after eating second day, leave it for cool it, leave it for 24 hours, heat it again on the third day. So if you eat it like this, whichever of the microorganisms which are in spore form might have turned into vegetative form, and when you eat, they will also get killed. So the intermittent heating and cooling technique to kill the endospores or resistant structure of bacteria is called as Tyndallization. So, I'm sorry, this is the experiment done by Louis Pasteur to identify the wine spoilage. I was not knowing this slide is there. So here he has taken the two wine sample, one is spoiled, one is good. So he has observed under microscope, he got two types of microorganism in spoiled wine and one type of microorganism in good wine. Then he has taken a fruits, grape juices in 3 flasks. He has heated them to kill all the organism. They inoculated the pure culture of all three. First one to this one, second one to second flask, third one to third flask. So he fermented. So when he is fermented, he got two good wines and one spoiled wine. And he identified that this bacteria, lactic acid bacteria, is responsible for sowing or spoilage of wine. And he also said yeast, this organism which he identified as yeast are responsible for production of wine or alcohol. Apart from that, there are other some other developments or discoveries made by Louis Pasteur. They are listed here. You can go through. Next, another important scientist in the field of microbiology and pathology who has contributed greatly for the development of microbiology is Robert Koch. Robert Koch is a doctor in England. He is an English physician. He is credited with the development of pure culture techniques. He is the one who has given the methods of separating microorganism from group of microorganism, that is called as a pure culture technique. He is the person who has first developed a solid media. So earlier what they were using fruit juices, carrot juices, or the blood samples, urine samples for growing the microorganism. So only liquid media were there, solid media were not there. So if there are solid media, the microorganism can form a colonies and they can be easily picked and put it in a new sterile solid media and we can get the pure culture. So he for the first time used gelatin as a certification, gelatin as a certification agent. So and he has also given different methods of picking the colonies and streaking. So streak plate methods for pure culturing of the microorganism. Later on, he has also used agar-agar as a certifying agent. So now also we are using agar-agar as a certifying agent to get a solid nutrient media. So and he developed a different streaking methods on solid media to get a pure culture. I was telling you, he gave the proof for germ theory of diseases. Louis Pasteur said that germs will cause the diseases, but he didn't say that this germ will cause this disease. But that is given by Robert Koch. Robert Koch gave the proof for germ theory of diseases. He said that Anthrax is caused by Bacillus anthracis. Tuberculosis is caused by Mycobacterium tuberculosis. Cholera is caused by Vibrio cholera. Like this. So he has identified that these organisms will cause this disease. And also he formulated, this is very important, he formulated Koch postulate. What are Koch postulate? He formulated four postulate, 4 point, that if you want to call any microorganism as a causal agent of particular disease, it should fulfill these four postulates provided by Robert Koch. That means, so if you go and see a diseased person, if they are having a particular symptom, okay, so if you want to say that if this person is having an anthrax or cholera, so if in a group of people there are four people having a symptoms, if you want to say that this is cholera disease, all people should show same symptom. The causative agent must be found in every case of the disease. All four people should have the same symptom. And when you take the sample from this blood sample, from this sample, and when you identify, when you isolate a causal microorganism present in that, so and the same causative agent should be present in all the disease specimen and should be isolated in pure form. And when you purify this organism from a diseased person and when you inject it into a healthy person, so the healthy person should produce same disease symptoms when it is injected the causal organism. And again, from the secondary, when you inject and the person get infected, so from that infected person, you should be able to isolate the same organism in pure form. Then it will prove that this organism is causing this disease. So there is Vibrio cholera will cause cholera, like that. I'll once again read Koch postulate. The causative agent must be found in every case of the disease. The causative agent should be isolated from the disease specimen and should be capable of culturing in pure form in laboratory. It must reproduce the same disease or symptom when introduced into a healthy organism. And same causative agent must be re-isolated from the inoculated host. So any microorganism to be called as a causative agent of particular disease should fulfill these four postulates. I was telling you that about pure culturing. Pure culturing, for example, you can see here, this is these are the colonies of bacteria that are grown on nutrient medium. So this individual colony are pure in themselves. So whenever a cell or a spore falls here, it will keep on multiply. It multiplies and forms a colonies. Similarly, these are the fungal colonies, maybe from soil. When you do the serial dilution and plate that, you will get different types of colonies. So from these are solid media. If solid media is there, we can able to get the colonies like this. But in liquid media, we cannot able to see a separate colonies like this. So how to pure culture? So you can select any colony. In case of bacteria, you can select this one, this one, this one. So without touching the others, in an aseptic condition in a laminar flow chamber, if you take this one and put it, streak it on a new sterile nutrient media. So after incubation, the growth, whatever you will get, it is a pure culture of this colony. Similarly, in case of fungi, so without touching the other colony, if you just take little bit inoculum with the help of inoculation loop and transfer it to a new plate, you will get a pure culture of this fungi. So this technique was given by Robert Koch. So another interesting story on the use of agar-agar. Actually, Robert Koch as an associate who was working with him. So he's called as Richard Petrie. He is the Richard Petrie. So, and Richard Petrie's wife name is Fenne Hesse. Fene-Hesse. So actually, she proposed the use of agar-agar as a certifying. Actually, Robert Koch initially he was using gelatin as a solidifying agent to prepare a solid medium. So in summers, what was happening is if the temperature goes above 35 degrees centigrade, this gelatin used to melt and become liquid. So it was solid only below 35 degrees centigrade. So that was the problem. So when Richard Petrie discussed this with his wife, so suggested one thing to use a agar-agar. So agar-agar, she had a relative in Japan. So when they went to Japan, she heard that and she came to know that agar-agar is used as a solidifying agent or a gelling agent in ice cream, salads, and other things. So in food preparation of some food. So she had bought some samples, some of around 100 to 200 grams of agar from there. So to prepare the salads and ice creams. So she suggested her husband Richard Petrie to try this one as a certifying agent. So he suggested this to Koch postulate, and when Koch postulates used agar-agar as a certifying agent, it worked wonders. So because even if the temperature goes up to 45 or 60 degree, this certifying agent was not melting and becoming liquid. So for the fact, you should know that agar-agar, once it becomes solid, it melts or becomes liquid only when it is heated above 95 degrees centigrade. And when it is cooled down to below 45 degrees centigrade, it again becomes solid. Okay. Robert Koch, I was telling you that he has given a proof for germ theory of diseases. Germ theory, so you know that Pasteur showed that microorganism made wine and beer go bad, food germ cause diseases, food microorganism cause diseases. Okay, so Robert Koch, as I was telling you that he has given a proof for the germ theory of disease. He identified through his Koch postulate that several organisms causing diseases. For example, he discovered the causal organism of anthrax, cholera, tuberculosis. So here, this is a list of diseases uh, along with their causal agents. For example, you can see anthrax caused by Bacillus anthracis, identified by Robert Koch. Tuberculosis, Mycobacterium tuberculosis, identified by Robert Koch. Cholera, Vibrio cholera, identified by Robert Koch. So like this, there are also several diseases, other diseases, causal organism, and the inventor for your reference. Now,